Okay, today we are going to start talking about protein structures. We have already made some introduction to protein structures at the beginning of this course in first week. And from time to time, we went to, for example, while we were talking about homology, I showed you some protein structure examples showing that structure is more preserved than a sequence. We have visited the protein structure data portal, the protein data bank, which was at rcsp.org.pdb. So this rcsp, the protein data bank, this is the main repository for protein structure information. In recent years, more protein structures are made available experimentally, and also we have recent developments in protein structure prediction, the deep pred by Google research that predicts by using deep learning methods. They were able to predict the protein structures with high accuracy, and especially this was apparent in the final protein structure prediction competition, CASP. The latest CASP contained this deep learning based prediction of protein structures, and organizers, the original organizers of CASP competition, they also stated that we can now consider this problem solved. Protein structure prediction was one of the unsolved problems. It was one of the millennium problems, and if you maybe remember, a couple of years back, maybe 5-6 years ago, there were these screensavers that the like community folding, you may call it. You were using your idle time of your computers to search the protein structural space for finding predictions for certain sequences. But yeah, with the availability of these deep learning methods, I think we can have more accurate predictions for protein structures. And also there are also developments in the experimental area as well, like cryo-EM, cryo-electron microscopy is one of the novel methods that also helps find protein structure experimentally. Now the good thing about cryo-EM is that this advantage over X-ray crystallography is that it may help measure the protein structures in their native state. So yeah, with this introduction, let's start talking about what protein structures are. And right now, if you take a look at this website, let me zoom in. If you look at the statistics, well, when you zoom in, the statistics at the top disappear. So yeah, it has to be at this zoom level. Around 177,000 protein structures are available today in Protein Data Bank right now. This number, as you can see, is very small compared to the available number of protein sequences. If we go to SwissProt or UniProt protein sequence database. So if you go to UniProt, it's another portal for protein sequences. And there are, in Tremble, there are 200 million automatically annotated but not reviewed. So there are 200 million protein sequences. Out of this 200 million, this 177,000 is still a very small number. So there are lots of protein structures with protein sequences with unknown structures. And SwissProt database, which is like roughly the 3 times the size of Protein Data Bank, contains manually annotated, which means that these proteins are protein sequences with known functions that are curated. But this is the protein sequence database. Tremble, on the other hand, contains 200 million protein sequences. So what this shows us that protein structure is difficult to obtain. You may obtain the sequence of a protein. So we have 200 million protein sequences. By using sequencing technologies, you can get the amino acid sequence of a protein, but finding its 3-dimensional shape is still challenging. The experimental methods we have, like cryo-EM, X-ray crystallography, nucleic magnetic resonance, NMR. Those are the three main methods to determine structure of proteins experimentally. And they are really expensive, and shows you that they're difficult to run, execute on these protein sequences, because there are less than 200,000 protein sequences sold experimentally these days. There are several biochemical difficulties. For example, I know that membrane proteins especially are very difficult to obtain. There's membrane proteins like 2 years ago. We had only a handful of membrane proteins with solved structures. Membrane proteins, their native state is in lipid membrane. So the membrane of the cell is lipid, not water. So it's not like other proteins in a soluble environment. Other proteins which are in the cytoplasm, they may be surrounded by water, but membrane embedded proteins in their native state, they're surrounded by lipid membrane. So they're very difficult to crystallize. In X-ray crystallography, for example, one thing that the researchers try to do is they put a certain number of these proteins, they crystallize them, and the crystal structure is used, and X-rays are shot through these crystal structures to get an idea about the three-dimensional shape. So that's something really difficult. In cryo-EM, with electron microscopy, which is able to give some, again, indirect measurements about the protein structure by using certain algorithms. These indirect data is solved to get the final three-dimensional shape of a protein structure. So let's take a look at this molecule of the month, this month in May, fetal hemoglobin. So this is the adult fetal hemoglobin, the hemoglobin of the fetus, and the adult hemoglobin. I cannot see any difference between these two structures myself, but yeah, they may be very similar. But hemoglobin is a protein structure composed of 4 chains. The chains you see here on the right-hand side. There are 2 alpha chain and 2 beta chains. They form 4 chain. When I say chain, a chain is a sequence of amino acids by amino acids connected by a peptide bond. So a single chain of amino acids is like a single sequence with a beginning and an end. The beginning is called the N terminus, the nitrogen, and the end is called C terminus, the carbon, because the nitrogen is free to accept bonds at the N terminus, and the carbon is free to accept bonds at the C terminus. So, and each successive amino acids are tied together in this backbone from nitrogen, carbon. We are going to see the details of that, but it's mostly the structure is lots of chemistry, biophysics involved here, and these are really big atomic structures. We have seen certain chemicals, the chemical compounds next to these protein structures, they are really small things. There are thousands of atoms. The molecular formula of a big protein structure like this contains thousands of atoms. So they're sometimes called macromolecular structures, but still they are at the atomic level. These are what you see here is a cartoon representation of the hemoglobin protein structure with these protein chains. These like spherical shapes, they are individual atoms, or maybe they may even be amino acids, I'm not sure. This is like a cartoon representation. We don't actually see a real picture. A real photo is impossible to take, because what you're seeing here, what you're looking at here, the atoms, their influence region, they're basically electromagnetic influences around them. They have no shapes, all right? So we cannot, their length is less than the wavelength of the light, which means that they are not visible. So all these things, all these physics make it really difficult to take a photograph, take a picture of of a protein structure. So that's why they're difficult to obtain. We use X-rays or other indirect methods, electron microscopy, for example, or nucleic magnetic resonance to get clues about these macromolecular structures. And a protein's shape is really important to its function. There are so its inside is not empty. There may be cavities, but it may be a tightly packed structure. But when I was talking about the protein chain as a sequence of amino acids connected to each other by peptide bonds, and this sequence of amino acids, they fold. So the protein folding problem is the finding the folded shape of this protein structure. Unlike DNA, DNA has a double helix structure, although it also folds onto itself around the histones. But here it has some regular shape. Here in protein structures, we have many different shapes. It's just the protein structural universe is really huge. There are different folds, different types of shapes that you may find in protein structural universe, and proteins are the building blocks of cells, and therefore they have different purposes. I have shown you this animation of kinesin walking on microtubules, right? If you remember that, let's go to YouTube. Yeah, this one. So these animations are, for example, this is a protein, its shape, how it carries something around. This is a microtubule. It's also a protein structure. Microtubules, and this is like a motor protein. Which interact physically again by using some biochemical reactions and some physical interactions between those protein structures to make things function in cell. And there is also the ATP synthase animation. Yeah, this is also nice. Look at this. These are really big macromolecular structures
in the membrane. There's the stalk, including the delta and the B subunits. The alpha subunits are shown in light pink. The beta subunits are magenta. In the middle, the central stalk is rotating. The B subunit stabilizes the hexamer, which means that the hexamer can't rotate. We will now look at a rendering of known structural states and the conformational changes that are happening. The beta subunits are seen as a ribbon diagram. The ADP and inorganic
this cartoon representation, when you look inside, this is also a cartoon representation of the same protein chain. You see certain regularities, certain local shapes. These red parts, which are shown like ribbons. Here, for example, they are called helices. Alpha helices are the most common helices in protein structures. These yellow parts, which are like parallel to each other, they are also tied to each other, and they are next to each other. They are arranged like in sheet formation on top of each other. These are called beta sheets. Beta sheets, alpha helices. There are hydrogen bonds between these adjacent beta sheets. And in the protein sequence, in the one-dimensional protein sequence, the amino acid sequence, these parts may be far away from each other. What makes protein structure prediction difficult is that parts that are far away, amino acids that are far away in sequence may actually be neighbors when the protein folds onto itself like this. Certain amino acids which are far away, we're like 100, 200 amino acids away from each other on the sequence, they may actually be interacting in the structure. They may have a hydrogen bond in between them. Helices, they have hydrogen bonds between every ith and I plus fourth. So every fifth amino acid is going to contain, so it turns around itself, and these two amino acids become neighbor with each other. So there are hydrogen bonds that make this helix also have this stable shape. So there are some regular local shapes, as you can see, helices, beta strands, like these coils or loops or turns. They're called turns or coils that connect these different local shapes. And this whole structure that you see here, its shape, it's also not static, as you can see. There are vibrations. When in its native state, certain parts of it are flexible. There may be small vibrations, small changes, conformation changes as it performs its function. And everything that governs this is physics, all right? So biochemistry, biophysics is really, but simple chemical formulas between small number of atoms is really difficult to explain these very large scale interactions between these thousands of atoms. Therefore, this is a really complex process that is taken on in cell. And the tools that analyze these dynamics of these things, how they interact with other chemicals, how they interact with each other, and many of these things, they happen in nanoseconds. They're really fast. Okay, so trying to simulate this thing, the research area that try to develop methods, tools to simulate the atomic level interactions is called molecular dynamics. So if you ever hear about molecular dynamics simulations, it will probably mean think about the interactions between protein structures, chemical compounds, and protein structures, and vice versa. So this one,
organic phosphate diffuse into the active site, and the ATP has just been released. And simultaneously, the conformational change occurs in the beta subunit. If we zoom into the active site, we can see the molecular structure of the substrates and the chemical reaction. Here is ADP with its two phosphates. They are all held in place by a network of hydrogen bonds mediated by the amino acids in the
usually in chemistry class, if you remember your chemistry class, you may think about a chemical reaction going on. Okay, on the left hand side there are some inputs and there's an output, and there are these actors in chemical reactions which are called enzymes, right? Those enzymes up to this point, they were always in the background, right? We just say enzyme. When you say enzyme, you may even have some, as an intuitive meaning, you may have something small as an enzyme. It's like it catalyzes the reaction. The actors that go into the reaction themselves are the main things, right? So one thing that is acting like an enzyme should be something small. But if you look at this picture,
the folded
this one here, this macromolecular structure, this is the enzyme, and the chemical reactions that are going on are between very small compounds. So the chemical reaction is possible. For example, binding of a phosphate to an ADP to synthesize ATP. This ADP plus P, the output is ATP. And we have an enzyme there. This enzyme is this big molecular machine. Okay, and the way it does this, let's again zoom here, is
active site, we can see the molecular structure of the substrates and the chemical reaction. Here is ADP with its two phosphates. They are all held in place by a network of hydrogen bonds mediated.
The enzyme, it binds to the ATP synthase molecule, which is the protein structures with A's at the end are protein structures that act like enzymes. That are enzymes. So what happens is that with these hydrogen bonds, there's a beta chain amino acids here. There are some free hydrogen bonds here, I think. And so this ADP binds
by the amino acids in the folded structure of the beta subunit. Hydrogen bonds stabilize the base and the sugar, but really primarily the base. There's also a magnesium ion that plays a critical role in not just stabilizing, but also neutralizing the charge of the phosphates. Notice the conformational changes that are happening when the inorganic phosphate comes in. There is the promotion of a planar transition state. Synthesis of a new phosphodiester bond is catalyzed, and then the subunit switches to the open conformation. Upon this reconfiguration in the beta subunit, ATP is ejected from the active site. The enzyme then resets and restarts the synthesis of another ATP molecule. This is a summary of the events at the active site. It is also intriguing just how the C ring rotates. We can start with the analogy of a water wheel. We can easily grasp the idea of how a water wheel rotates. It's driven by the flow of water, allowing the wooden fins to go in a particular direction. Thus, the rotation happens in a specific direction. If the water was to flow in the opposite direction, you would see rotation of the water wheel in the opposite direction. Here we see the C ring and the A subunit. We don't know the full structure of the A subunit, but we know that there's an access channel for protons on the A subunit right in the intermembrane space. And there's a separate egress channel for protons on the matrix side.
So I'm not going to go into details of physics, chemistry of this thing, because it's really complicated. It's outside the scope of this class. Just know that at this point, protein structures are really big molecular machines, and their structures are important. The amino acid sequence we have seen up to this point, it's really what makes that amino acid sequence. Those amino acids, they fold onto each other, form these three-dimensional shapes to function. Okay, so let's continue with this slides after the introduction. All right, and I also want to show you in this PDB data bank. Well, so it was let's, for example, search that ATP synthase was a nice molecule, right? So if we search for ATP synthase in protein data bank, its structure is known since 2005. So if you click on it, there are different big rotary motors, motor to generator, parseless. It's just like an engine. Right, it's they're parsed, the F1 structure. So this PDB entry 1E79 includes the F1 motor of ATP synthase. So if you want to get this rotating part F1, you can click on this PDB ID 1E79. PDB IDs are 4-letter IDs. They contain numerical values and some letters, but they're 4 characters long. And let's click on this PDB ID, and it's going to get to our page. If you look at this, the biological assembly of this 1E79 is the bovine F1 ATPase inhibited by DCCD. I don't know what that is, but if you look at the 3-dimensional view interactively, with the help of web technologies, we can also interactively visualize them on our web browser. Look at this. So this is that protein structure. So this is the cartoon representation. So the way we visualize this is basically an artistic drawing. You may consider this an artistic drawing. You see the helices, you see beta strands, and you can actually, while you're viewing this, you can even highlight individual amino acids, I think, along this chain. So this is the chain of, which chain is this? This is the chain A. So chain A, which one is chain? Yeah, I think this green one is chain A. As I move my pointer along this, as you can see how the amino acids are highlighted in the sequence. You can also do vice versa. As you move, I think it will highlight. Yeah, it's highlighting. So this leucine that you see as you move along, you may see right at this point. If you focus at this, yeah, if you look at this point, how protein structure, these are individual amino acid. An amino acid contains 10 to around tens of atoms in it. So there are 10 to 20 atoms there are here in this region. So we can also view it with a different, so we can zoom in, zoom out, rotate this, and there are multiple chains. This is the C chain, A, B, C, different chains. We can visualize their sequences. Components, ion density. So how do we, oh, so okay. Layout, moving in background, mass controls. This is not what I'm looking for. Well, you can zoom in individual. So, or you can Yeah. When you zoom out, for example, there's also like far field that is cut off as you move away. So let's get back to the normal thing. I just want to show you different ways to visualize this. Structural motif search measurements, type assembly, export animation. Yeah, oh, here, right here. This is the cartoon-based representation. I think I'll be able to change this. Edge representation. Yeah, cartoon. So if I do balls and sticks, I can also view the individual atoms as common chemical type of. I think I should also you just add representations as you go there. Can you delete representations? No, these are when I say cartoon. Oh, there are 3 representations now. Yeah, the interface is not very easy to use. So you can add representation, but how do you remove? Oh, okay, it's right here. So the representations, so you can actually hide the cartoon representation. This is the balls and sticks representation, which shows the same molecule. Now you see it's viewed in a different manner. So let's hide this as well. Now let's say edge representation, Gaussian surface, molecular surface. So when you say molecular surface, I think it's currently building the molecular surface. Yeah, this is the molecular surface of the same. It looks more like the one in the animation, right? So molecular surface is the solvent accessible surface that we see here. Only the outside is shown to us. So they're tied together in this nice Play-Doh type of shape. So this molecular surface representation is used a lot by molecular biologists. But if you want to see the details inside it, you can you can get other representations too. So if you erase this, let's see what else do we have. Point space fill. Yeah, this one is going to show the atoms in it. If you want to see, can a particular protein sequence have, that's a very good question. So a particular protein sequence can have more than one possible shape because the shape sometimes is defined also by some external factors. For example, if you put the same protein inside, not inside water, but inside lipid membrane, it may look different. Or if you take a membrane embedded protein in its native site, okay, which is in lipid environment, if you put it in water, it may make it take a different shape because these amino acids, they have different physiochemical properties. For example, if this is a soluble protein, if it's inside cytoplasm, which is if it's surrounded by water, the surface amino acids are going to be amino acids which we call hydrophilic. They love to be around water. Hydrophobic amino acids, on the other hand, which don't want to be close to water molecules, they will try to stick to each other and be at the center, at the core of, just like penguins avoiding cold in South Pole. Uh, uh, so the amino acids that hate water, they cluster each other, they stick to each other and make the core of the protein. So your question whether protein sequence, the same amino acid sequence, can have different shape, it is possible based on environmental factors, but most of the time a sequence determines the structure in terms of, I mean, there will always be exceptions. In molecular biology, you cannot have this because it's a really huge complex system. There are exceptions to everything. But you may consider if we know the sequence, it will have some structure. So that's unique, and you may consider that structure unique. So this statement, it will be true for maybe 99% of the protein sequences. If you know the sequence, it will have, it will fold in cytoplasm, it will fold into a unique shape. It doesn't change, it's really robust. It doesn't change quickly to a different shape. And this shape that we see here is static, as you can see, but when it's performing its function in the cell, there are certain vibrations, small conformational changes here and there. So whether it's currently active, whether, for example, like when a compound binds to this, it may, for example, it may change. If it may be like this, and after something binds to one place here, it may have a different shape like this. There may be small angular changes, so it's really dynamic. But the overall shape that we see here, even this static shape, is really helpful in understanding what is going on here. For example, I want to show whether you'll be able to see the electrostatic potential. So this one is just showing the coloring here. Each atom, first of all, I think there is a color scheme for every chain. In order to be able to see every chain differently, each atom in different chain is given a different color map. But within the same chain, there are also atoms are represented in different colors. So this is lysine in C chain. So there are leucine. So this leucine, and maybe not all the atoms are shown. The hydrogen atoms, which are really small, sometimes it's not shown. So then I say add representation. Let's see whether I have the electrostatic potential in molecular surface. What is this Gaussian surface? Let's try that. Oh, it's also similar to the Gaussian surface is also like molecular surface, but I think it's very much simpler to compute. It's a more crude representation of the molecular surface. It's not the exact molecular surface or solvent accessibility surface, but therefore it was really quick to obtain this. But I want to show you in this PDB website, I want to show you something else. Okay, let me zoom out again. Wow, zooming out. So let's go back to, yeah, this one. The good thing is that you can download the structure. Just like, for example, if you, this is like a three-dimensional file, right? If you're using a three-dimensional model of something, you would want to get what is the Blender file, 3D Studio file, 3D Studio Max format. So what I mean is that if you go to a place like Thingiverse and you want to develop a three-dimensional model of something digital, for example, like this rattlesnake. This rattlesnake is a three-dimensional shape. Okay, so when you try to download it, this three-dimensional shape, you're going to be presented with triangles, edges. So it has a geometry. Just like that, our proteins, this three-dimensional protein, it has a geometry, and you can download it. So PDB format is the most widely used format for representing protein structures. So if we download this PDB format, let's see how it looks like. So it's a text file, and it's been around, this format has been around before we had XML, any structured way, or JSON. It's really unstructured. It's a really old file format, in which every line contains some information, and it contains some keyword at the beginning. So for example, the header is hydrolase. So this 1E79 is also known as hydrolase. This is its title. This is the source organism. It's from Bostaurus. Okay. Organism's common name is bovine. Taxonomy ID. So this ATPase, for example, comes from the heart of a bovine. It's a muscle tissue. Since mitochondria is responsible for energy production in the cell, so the ATPase was synthesized from the mitochondrion. And when was this done? Let's see. 2014. So there was some revision. So in 2011, I think this is the first, or this is still the revision, I think. This is the paper. So it appeared. So this is, look at this, in the PDB file we have a metadata referencing to a journal, starting with this journal keyword saying that, okay, these are the authors, this is the title of the journal, the structure of the central stalk in bovine F1. So this was a journal in Nature Structural Biology in 2000. And they get this at 2.4 angstrom resolution. So these usually X-ray crystallography files, they come with the atomic resolution saying that what is the amount of error you can expect at individual atoms. So 2.4 angstrom is a really nice high resolution. And the important part, the part that I want to show that contains geometry information here is, let's all pass these remarks. Lots of comments, remarks about this. Different. I'm just skipping those remark parts. These are all metadata. And this is the sequence is given to us. The amino acid sequence is given to us as 3-letter amino acid codes. Here, these are individual lines that we see here. I think this is the total length of chain A. It contains 510 amino acids. And this is the sequence of amino acids from N terminus to C terminus. This is chain B. It's also 510 amino acids. They may be identical, meaning that this is called homo. It's not a dimer, trimer, homotrimer. If it's 3 chains coming together, if it's called trimer, is it called trimer? Does any molecular biologists know that? I know 4 of them are called tetramer, and 2 of them called dimer, but 3 of them should be called trimer, I guess. So, oh, and this is there's also the D chain. Okay, A, B, C, D, E chain. An F chain with different number of amino acids. So it's a really big protein structure with a lot of chains. So this is the sequence. And here's we don't see yet the geometric information about this protein. There are some local structural shapes that are known, which are also representative. There's a helix between this and this. So some helix information, beta sheet information. So local structure information is also given in this one. Here, these are the records which comprise the majority of this file. So this file is, let's see, the size, 2 megabytes. Okay, it's a text file, 2 megabytes. How many lines are there? Let's see. It contains around 28,000 lines. In those 28,000 lines, almost all of the records are atom records. Okay, the atom records comprise most of the file. As you can see, most of it is atom. So what does this atom record tell us? That's where the geometry comes into picture. I mean, you may also say that the entire PDB file is just, if somebody gave you just the atom records, that will also be the minimal PDB file. And there are some tools or methods that work on protein structure which only look at the atom record with the special CA at the 3rd column. CA means the alpha carbon of that amino acid. Every amino acid contains these 3 backbone atoms: nitrogen, carbon alpha, and the other carbon. This is carbon beta, oxygen. And look at this PDB file. There are no hydrogen atoms. Hydrogen atoms are they're small, and some PDB files will not contain the coordinates for those hydrogen atoms, but they can be modeled if you know the positions of these atoms. So at the atomic level, this PDB file gives us the X, Y, Z coordinates for the center of that atom. So atom is not, as I said, it's like atom's position. I mean, is even not certain, but with very high resolution, we based on its influence area. So the atom has been given a position by this structural modeling, structural determination. Okay, and this tells us, for example, the fourth column tells us that these 1, 2, 3, 4, 5 atoms are part of the first amino acid, alanine. This is either aspartame or asparagine. I don't know. I don't know the names of the amino acids well. So it also it contains more amino acids, as you can see. THR. This is probably serine, valine. Okay, as you can see, there are different atoms specific to. Some of these atoms are specific to certain amino acids. They don't contain every time. For example, this alanine is probably smaller than this other amino acid. It has more oxygen. OD1 and OD2, we don't have it in this one. But the really nice, remarkable thing is that if you know OpenGL, if you're our department student, you have taken computer graphics probably the previous semester, you can just write your own visualization tool in OpenGL by putting a nitrogen sphere centered at this XYZ coordinates. So these are the XYZ coordinates. I don't know the detail of these parts, so this is the atom name at the end. There are some, this is like temperature, or I usually ignore these parts. The important part is this XYZ coordinate, but you can have your own visualization tool by just putting spheres into these XYZ coordinates, and you may use the van der Waals radius of these atoms. How big is a nitrogen atom? How big is a carbon atom? Nitrogen, carbon, oxygen. These are the main atoms that we see in these amino acids. There are other atoms like 2 amino acids. They also contain the sulfur. The cysteine and methionine, I think, also contains sulfur in it. So, but yeah, look at this. Thousands of atoms. And these are atom IDs. Let's see how many atoms we have. And ignoring the hydrogen atoms. Ignoring the hydrogen atoms, this is atom number 1. Oh, by the way, this is like amino acid number, amino acid 19. It tells you that we don't have any affirmative position for the atoms of the first 18 amino acids. So the first 18 amino acids of this chain has an unknown position. So the accurate positioning could be obtained after the 19 amino acid. I think this is like the 19 amino acid in the chain. It's the amino acid ID, but each atom is also given a number here. In this first chain, let's go all the way to the end of the first chain. Right here, yeah, there are close to 4,000 atoms, ignoring hydrogen, in the first chain. So if you combine all these different chains, actually at the end we'll see this is incremental, the atom numbers. Yeah, right here, 25,000 atoms. So if you were to write an OpenGL program to visualize this protein structures as balls of atoms, spheres, you would have 25,000 spheres in your visualization. So in other words, what we have seen here, when we have looked at the structure of this one, and when we have seen it in this ball and stick. So let's also hide. So there's some water and ions around this. I'm going to hide them. And I'm going to say add representation. And when you say the space fill, let's hide that. So this space fill representation that you see here, you see that many. If I mean, there are like 25,000 spheres. If some of them are inside, we don't see all of them, but you see how remarkable it is. We can interactively visualize this many spheres. When I started my PhD back in 2000, this was one of the first problems that I attacked in my PhD studies, how to interactively visualize this many spheres in Java 3D. There was Java has a three-dimensional library other than OpenGL because Java. I mean, now we can visualize this on the web browser. Back then, web browsers did not have this three-dimensional visualization capabilities, but with HTML5 and with WebGL, it allows us to interactively view this many structures, and with the also advancement in graphics, GPU technologies, we can visualize very large number of spheres with very high frame rates interactively. So this is really remarkable. Okay, so yeah, these are three-dimensional coordinates. So I think this was a good introduction to protein structures. What we are going to see before I end today's lecture, I want to just, so this was just a general introduction to protein structures. I will go over these slides again on Monday. But what we're going to, the problems, the computational problems that we're going to talk about protein structures, there are two main problems we will discuss. First is protein structure prediction. If somebody gives us the sequence of protein sequences, how do we predict which three-dimensional shape it's going to have? I'm not going to go into details of methods, but I'm just going to talk about in general, the general approaches researchers use to tackle this problem, like homology modeling, which makes use of sequence similarity. If another protein structure with known shape has a sequence similarity to this target sequence, they may also share some structural similarities. That's called homology modeling. There is the approach of threading, which I call the Cinderella method, which tries to thread a sequence onto a known shape to see whether that sequence fits the shape physically. We are going to talk about threading. There is also ab initio modeling, which tries to use chemical formula, physical associations between individual atoms, and try to solve the structure by using no previous information about other known structures. So these are the three methods that we're going to see. And also, I'm going to talk about structure comparison problem. If I give you two structural protein structures, two shapes, okay, these two protein structures, in terms of shape, how can you align them on top of each other? So this is different than ducking. In protein structures, there's also the ducking problem. When you give two protein structures, whether they're going to fit each other nicely, that's called the ducking problem. We're not going to discuss this in this course. The protein structure alignment or structure comparison problem is unlike ducking. It's not trying to find complementary shapes. Given two protein structures, it tries to identify whether are there substructures, are there parts in these proteins that are similar in shape. As an analogy to sequence similarity, we were looking for amino acid sequence parts that are matching between two sequences. In structural alignment, we will be looking at whether there are shapes. So this is basically a shape matching problem. Whether there are certain substructures, shapes in these proteins that are similar to each other in terms of their geometry. That's called the structural alignment problem. That's also a problem that I studied during my PhD. So I studied protein structures in my PhD studies. So I also proposed a method for protein structural similarity, but there are very well-known methods that are in use today to find structural similarities between proteins. So these are the two main problems: structure prediction from sequence and structural alignment between two protein structures is the problems that we will discuss under the topic of protein structures. All right, so that's it from me today. I will be happy to answer if you have any questions. I will stop recording now.